Well, student, a GMO is any type of organism that has been genetically modified, hence GMO. And this change has to come from humans and not from any mutation that has naturally been done. Well, it is a highly scientific process and occurs in 3 steps: electrophoresis, recombinance, and cloning. If scientists want to genetically modify an organism, they first got to find the gene that causes the trait that they desire. So they go through a process called electrophoresis. Now, in electrophoresis, scientists fill a bin with a solution of agarose. Agarose, when it dries, is very porous. And after they let the agarose dry, they fill it with a buffer solution that fills in these pores. And since DNA and RNA and other proteins are negatively charged, they put 2 electrodes on either side of the bin and they conduct an electric current. And this current causes all the DNA to float to one side. And since DNA can be of varying sizes, smaller bits get farther than longer bits. Well, recombinance is the step that scientists take to insert a gene into a circular piece of DNA from bacteria called plasmid. And plasmid has 2 important zones. One of the zones is the ampicillin resistance, which is an antibiotic that the bacteria can come across, and the lac-Z region, which is a region in the plasmid that has a large number of restriction zones. And these restriction zones are able to be cut by an enzyme. And what this enzyme does is it comes in at these specific restriction zones, which is dignified by certain codons. They come in and they take this, just imagine this as a part of the DNA, and they take the phosphate bonds right here and they rip them at one end and then also at the other. And since the bond in between the nucleotides, called the hydrogen bond, is very weak, these can easily separate. So we just have one long strand of DNA that has sticky ends that are positive and negative. So after they have mixed all the enzymes in with the plasmids, they take the desired trait and they also mix it in with them. So when you mix them in, most of them will probably not mix with the right trait. They might stick back together, but you will have some plasmids with the trait encoded into them, which is something that we want. Well, my very attentive student, the only reason they stick is because of electronegativity. Since that region of the plasmid has been cut by the same enzyme, one end is either a negative or positive with an electric charge. And also the trait that we wanted has also been cut by the same restriction enzymes. So when they're mixed together in the solution, the negative side will stick to the positive side, like so, and then the same thing will happen on the other side of the genes, so it completes the circle again with kind of like a little triangle on the side. Well, the 3rd step in this is when scientists try to get the gene to be expressed. So since they have all the plasmid and traits into that one test tube, they'll also add bacteria after that. And after they add the bacteria, they'll do a process called electroporation. And this is when they send quick and small electric pulses into the test tube, which will break up the cell wall of the bacteria and let all of the plasmid carrying traits into the bacteria. And since not all the bacteria will get the plasmids with the desired trait, they are transferred into a petri dish and they are filled with ampicillin. And ampicillin, if you remember, is the antibiotic that a plasmid carries the gene to be resistant to. So only the ones that have the plasmid will be surviving after that time. So then after that, they will culture the surviving bacteria, and since this might take a very long time, they use hundreds and hundreds of samples just to get the one that they want. And after they get the one that they want, they culture it even more. Well, an example of a GMO is an Enviropig. An Enviropig is a Yorkshire pig that has E. coli phytase gene and a murine-imperated secretory protein gene that was inserted by pronuclear microinjection into a fertilized porcine embryo surgically implanted into an estrus synchronized sow. So the first part is the E. coli phytase gene. And this gene is found in E. coli that produces phytase, which is able to digest a form of phosphorus called phytate, which is found in some of the food that the Yorkshire pig eats. And the second part is the murine parotid secretory gene. So murine has to do with anything about mice, and the parotid gland, which is the second part, refers to the glands that is found right above your ears that secretes most of your saliva. And secretory just means anything that will secrete. So this gene is formed from both E. coli and mice. So that's pretty cool. So what it does is lets the pig secrete phytase, which will then break up phytate that it digests. Alright, for the second part on how they got it into the pig was the pronuclear microinjection. So what they basically did is they took an egg of a developing piglet that does not have a nucleus yet, so all the chromosomes are already just wandering around, and they injected it into there before the nucleus had formed. And the second part was that it was inserted into a porcine embryo. Porcine means anything that has to do with pork. So they're just talking about pigs right there, and they surgically implanted into an estrus synchronized sow. And what this means is they cut open a mama pig that was ready to carry a little piglet, and they inserted it into her uterus. The three reasons that they make an Enviropig is, one, because of the phosphorus they take from phytate will transition into less supplement needed and less cost to make those supplements. The second reason is because the manure will be lessened because less phosphorus will be wasted. And the third is because of runoff from that manure, and that will get into the water streams and kill fish because the phosphorus will bind to oxygen and make it useless for the aquatic life. The two drawbacks of this organism is that it can only be done in Yorkshire pigs, and that so little is known about the modification itself that the FDA and anybody else may not even approve it for years. How to improve this organism is by inhibiting the myostatin, which is the enzyme that regulates how big your muscles get. So if we implant this in a pig embryo, we can make a bodybuilding pig that thrives in the environment. Let's remember what a GMO is. So a GMO is a genetically modified organism, which is an organism that has its genes tampered with by humans to make a desired trait. And just to think about the Enviropig, can you imagine what we could do with other animals so that we can help our environment more, since we're kind of destroying it?